This protocol is significant because we can control the 4-electron reduction of CO2, and we can use the product form as a reactive and versatile intermediate in one-pot cascade reactions.
Thanks to the controlled CO2 reduction step and cascade strategy, this technique enables probing off the intermediate reactivity, leading to the synthesis of Cara compound from CO2 for the first time.
The reaction mixture preparation is a meticulous process that involves multiple additions to a reaction vessel inside the glove box, CO2 pressurization, and a strict time and temperature control.
This cascade strategy requires subsequent reaction steps, the use of a glove box, and gas handling, all of which are easier to understand through visual demonstration.
For synthesis of compound 2, charge an NMR tube with 15.9 milligrams of 9-BBN, 130 microliters of a 10% molar hexamethylbenzene as an internal standard, and 100 microliters of a 1% molar iron catalyst solution in deuterated tetrahydrofuran. Add 0.37 milliliters of additional deuterated tetrahydrofuran to the tube before closing the tube and connecting it to a gas system outside of the glove box. Place the NMR tube in a 25 degrees Celsius bath for 15 minutes. Then add 1 atmosphere of carbon dioxide for 3 minutes before closing the tube for a 45-minute incubation at 25 degrees Celsius. During the equilibration, prepare a stock solution by mixing 189 microliters